Hello, everyone. My name is Jenni Kovach, and I am the president of the EHE Foundation. I am also an EHE patient, so I am double excited about today. The EHE Foundation is a US-based nonprofit organization that was formed in 2015 by a dedicated group of volunteers. We have one goal: to find treatment and a cure for EHE. Together with the partnership of the EHE Rare Cancer Charity UK and the EHE Rare Cancer Research Foundation Australia, I'd like to welcome everyone to the 2022 EHE 360 Scientific Symposium. This is a two-day series. Today is the Scientific Symposium and tomorrow is the Patient Conference. A special thank you to our conference co-chairs, Dr. Rubin, Dr. Stacchiati, and Dr. Tapp. Thank you so much for providing your time and insight into helping us craft this conference. We truly appreciate you. Today, we have three goals: educate, innovate, and collaborate. And so with 337 total people attending over the next 2 days and 146 registered today, I am absolutely sure that we will meet these goals. So just a little housekeeping to get us started. You are currently on the 360 event site. Please feel comfortable looking around the site. If you scroll down, you're going to see the list of sessions. And then it will also show you our speakers. It will show you our sponsors. And at the bottom, there are a couple of, I guess, a handful of videos from our community. Please make sure to take a moment and watch those videos. Across the top, you're going to see a link that says attendees. I would encourage you to click on that link and a pop-up will come up saying, would you like to make your profile visible? Please do so, because this is how other people in the conference are going to be able to chat with you. If you need any help at all today, click on attendees and search for Maida Diora Sutliff. M-E-D-H-A. Maida is our executive director of the foundation, and she is available all day to help you out. So most of the sessions today are going to be a panel discussion, immediately followed by a moderated Q&A. So once you're in a session, you're going to see the Q&A function on the right-hand sidebar. Make sure just to engage and ask questions as they come up today. So talking about Q&A, let me introduce you to our moderator. If you were in attendance at our last year's conference, you may remember David Casimir. David is a board member of the EHE Foundation, and he brings so much expertise to the EHE community. I am thrilled that he will once again be guiding us through our Q&A portions of the day. Okay, so today is interactive. There are so many ways that you can jump in and participate. So please don't hold back. You can use the chat to comment. You can ask questions by posting it in the Q&A sidebar. You can message the other attendees directly. We have a feedback button on the homepage. The only way we can make these conferences better in the future is to hear from you. So please leave your comments. And then please just, if you post anything about today, let's make it social and hashtag EHE360. I do want to just very quickly let you know that we have a collaborative roundtable discussion at the end of the day. It is worth your while to stick around for this. We will be moving to a Zoom collaborative session and it will be open mic, and I'm super excited just to hear the conversation flow during this time. All right, before we get started, a quick thank you to our sponsors. Our lead sponsor for this event is Ikena Oncology. Our other two sponsors are Every Life Foundation and the Cleveland Clinic Sarcoma Program. And I also want to mention Chan Zuckerberg Initiative Rare as One program, who has been so supportive of the EHE Foundation. All right, let's get this party started. It's my distinct honor to introduce our first speaker, Dr. Brian Rubin, who is located at the Cleveland Clinic. Dr. Rubin is a celebrity in the world of EHE. Many of our patients, including myself, think of him as our #1 advocate and a dear friend to our community. Dr. Rubin will be providing a symposium opening address on EHE Biology, What We Know. Please welcome Dr. Rubin.
Thanks very much, Jenny. It's a pleasure to be here today to speak with a group of scientists who are interested in EHE as well as the EHE community. And it's really fun to watch this group grow. I have to say, when we started this project about 12, 13 years ago, I never kind of envisioned that it would end up like this, but obviously very delighted that it has. Let me share my screen. Hopefully this works. Okay, right now you should be seeing my screen. If anybody cannot see it, feel free to chime in and let me know that that's not the case. But as mentioned by Jenny, I'm Brian Rubin. I'm the chair of pathology at the Cleveland Clinic. And I've had a long-standing interest in the biology, genetics of sarcomas. And recently, over the last 10 years or so, we've embarked on this project with EHE. So my task is really just to kind of tell you a little bit about what is known about the biology of EHE. I'm not going to go into the clinical aspects of it at all, and I hope that most of us already know that, but there'll be a lot of people will probably over the course of the morning remind us of some of the clinical features and whatnot. So let me go to the next slide. All right, so this was the first thing we learned about EHE. I mean, beyond its description, was that it had a relatively normal cytogenetic profile. We see here 22 pairs of autosomes and a pair of sex chromosomes. This is a karyotype from an EHE patient published by a giant in the field of cytogenetics, a woman named Julia Bridge, who's formerly at the University of Nebraska, more recently down in Arizona. And what Julia showed is that there was a translocation involving chromosomes 1 and 3, the short arm of chromosome 1 and the long arm of chromosome 3, and no other genetic alterations detectable at the cytogenetic level. So, we learned that this is a simple cancer with a reciprocal translocation, and over the course of years, there were 4 karyotypes that got published, and they all had the same karyotype. So we knew that in essence, this was going to be characteristic of this disease. Then later, in about 2011, our group, and then I do want to mention that Christina Antonieska's group, I've cited her paper down at the bottom of this, we really published this story simultaneously. What was nice at the time was that we arrived at the same conclusion through entirely different methods. We were an early adopter of NGS-based genotyping, essentially, to determine fusions and things. And kind of everybody's now at the field, but Christina was using more traditional techniques that we used in previous studies and previously in our other work, using traditional fish and cloning kind of the old-fashioned way, which was actually really difficult. The way that we cloned the gene was quite easy. We used NGS. And so we identified a disease-defining gene fusion in EHE. And the fusion was what was described by that cytogenetic alteration. We published that with Science Translational Medicine in 2011. So when you look at the protein composition of that fusion, here it is. And so it fused 2 things that I knew little about at the time, but now I know a bit more about. One protein, or at the genetic level, the gene called TAS, also known as WWTR1, to a much bigger protein called CAMTA1. And we didn't know a heck of a lot at the time, but essentially TAS was an end effector of the hippo pathway. CAMTA1 is still relatively unstudied, and not a lot's known about it. It mediates drought tolerance and arabidopsis, thought to be a tumor suppressor in some glial things. It's really not expressed in normal human tissues outside of the brain. So kind of an interesting protein, but importantly for the biology of EHE, the T binding motif of TAS is retained. This is the N terminus of TAS. So we showed pretty early on that, in fact, TAS-CAMTA1 was transforming in NIH3 cells. In other words, we could get them to grow as colonies in soft agar. And this gave us a tool to then start to look around at and start playing with the protein and try to understand. And we published a paper in 2016, and Munir Tanas, who was in my lab at the time as a postdoc, now having his own lab in Iowa, will speak, and he'll speak tomorrow, led this work to show that TAS-CAMTA1 was transforming. You can see in the bottom panel here that we get colonies, we use NRAS as a control, and then we quantitated that over on the right. So it was quite transforming, and it was an easy assay to use. So that we knew that TAS-CAMTA1 was transforming. Now, the Hippo pathway became something very important to our lab. And so for those of you who don't know the Hippo pathway, it's a serine threonine kinase cascade with various inputs. We're not going to talk a lot about inputs today, but essentially different signals come down mostly through the cell membrane to MST1 and 2, then LATS1 and 2. Those are both serine threonine kinases. And their job is really to phosphorylate TAS and its paralog YAP, which are the end effectors, they're transcription factors. And phosphorylation is a negative regulator of TAS, so it causes it to be degraded, retained in the cytoplasm. And if it's in an unphosphorylated state, it migrates into the nucleus. And we're going to talk a little bit today about targeting. And, the kind of idea I think behind this talk was, what are the particular targets that we might start thinking about in this disease? And so one critical interaction that was ironed out well before we got involved in the story was between TAS and another group of proteins, TEAD1 through 4, that are proteins that bind to TAS and YAP through conserved sites and tether TAS or YAP to DNA and activate transcription, resulting in pro-growth signals, tumors, etc. This is actually from the original description, I believe, of TAS being cloned by DJ Pan, a real giant in the field down at Southwestern now. And, again, just to emphasize, this is a pro-growth pathway. So when TAS and YAP are not inhibited, they lead to growth. And in fact, the way he identified TAS, as you can see over on the right side of this screen here, panel B, that is a mutant Drosophila flyhead. And, it's got these overgrowths, and it looks very hippo-like. I think the name is fantastic that DJ came up with, and I think Jerry Rubin was involved too, because he was in Jerry's lab. But you can see a normal flyhead on the left, the hippo pathway fly on the right. So when components upstream of TAS and YAP are essentially inactivated, you end up with a phenotype, or you can activate TAS directly, or TAS or YAP, and you end up with a flyhead that looks like this. The wings, we call it a wing disc of the fly, is seen below here with a wild type little kind of ditzily looking thing on the left, and then the giant hippo wing on the right. Okay, and then we showed, again, this is Munir's work when he was in the lab, that again, the normal hippo pathway is anti-proliferative. So in this case, it's inhibiting TAS in its normal function. And when it's no longer inhibiting, when there's a reason to grow and for organ development or cells to grow large or whatever, then TAS works its way into the nucleus and binds TEAD. But what happens in EHE is that the binding of TAS-CAMTA1 allows TAS-CAMTA1 to spend most of its time in the nucleus. So it's not really regulated in a way that's very powerful by the hippo pathway. And this is still an area where we don't fully understand. We originally had thought that hippo didn't really interact at all with TAS-CAMTA1, or at least not in a productive way. But I think there's some more recent evidence that indicates and suggests that, in fact, it may still retain the ability to regulate. And so that would be one area, for instance, where you might start thinking about the ability to target upstream regulators of the hippo pathway. And I know that John Lamar, for instance, is interested in that area therapeutically. And again, we showed very powerfully, I'll just show you a couple panels from one of our papers. This is from our paper that we published in Oncogene, that in fact, when you ablate a single amino acid in TAS-CAMTA1 right at the TEAD binding site, and we knew this from other work that this was an absolutely critical alteration, that you ablate all activity in TC, TAS-CAMTA1, its ability to transform NAH3T3 cells or grow as colonies. And so we knew that this interaction was very, very critical. And you can see no colonies here on the plate. Then over on the right side, you can see the quantification where there's a 100% reduction in ability to cause colonies in soft agar. And then on the next slide, you do the cognate kind of experiment where you take down T using shRNAs, that you see exactly the same result. That in fact, you get no colony growth when you completely ablate T. And so just again, I'm just emphasizing that the interaction between TEAD and TAS is absolutely critical. And you'll hear a guy that's working in my laboratory currently, Ajay Pavati, is going to present some data later talking about how our strategy to look at that. And I think from what I know, every company that's involved in trying to inhibit the Hippo pathway is looking at that TAS-TEAD interaction as the place that they want to place their therapeutic strategy. So in other words, developing molecules which inhibit that interaction. We also did some other work in our laboratory looking at signal transduction. So early on, we thought it was going to be very tough to target TAS-CAMTA1. And so we tried to look at signal transduction known molecules to try to see if there was a place where we could inhibit. In fact, so what we showed is that I'm just showing CTGF here because it is a critical mediator. It's expressed as one of the targets of TAS-CAMTA1. It's strongly expressed in EHE cells. CTGF is made and secreted out into the cytoplasm where it binds to integrins on the cell surface of EHE. So it's feeding itself essentially in an autocrine manner, stimulates RAS and then MAP kinase pathway. And we actually looked at trametinib, a drug made by Novartis, as a possibility for a clinical trial. And we, in fact, executed that clinical trial. So another therapeutic strategy based on the biology is to look at downstream signaling, downstream of TAS-CAMTA1 activation. And we focused on MAP kinase. We did a study, and Scott talked about it last year. So this was the first prospective clinical trial in this disease. And for those of you who are from drug companies, this was a very successful trial. We ended up recruiting 27 patients, and the trial is essentially finishing up now, but we were able to recruit pretty quickly. And so in this disease, doing focused clinical trials is a possibility. And it's something that I think that we certainly could do when interesting drugs come to the fore. Okay, and then finally, and most recently, we just published this paper last year. And really what we showed, in fact, that you can express TAS-CAMTA1 in mice through, and I'm not going to talk about this, but somebody from our lab is going to talk about the latest iteration of this model. This is actually a more, a model that we published last year. We have a sort of a new and improved version of this, this model, but essentially what we showed is that you can express TAS-CAMTA1 in the appropriate cellular context, so in endothelial cells, endothelial cell precursors. And what you end up with is a model which absolutely recapitulates EHE to a T. The cells, the lesions look exactly like EHE. They have the same expression profile, et cetera. And so it's a model. But what was really fascinating was you could take a single gene alteration and you get the disease. And we knew that from genetics in humans. And so this is just the last slide I'm going to show. And this is actually published by James Chen and his group down at Ohio State, but it's just data from Foundation Medicine. So they actually just summarized the Foundation Medicine data and published that. So there's 49 EHE patients, and importantly, 94% had the WWTR1 CAMTA1 gene fusion. So it's a really consistent alteration in this disease, and it's really disease defining as we originally published. Then only 50% or so. So that and that was the only finding in about half the cases. So again, kind of supporting why we even made the mouse model. You only need that alteration to get the disease. Now, to get what we think are more aggressive forms of the disease, you need to start knocking out or activating other genes. But the repertoire of what happens is pretty simple. There's very few alterations secondarily, and only about half the cases have these. And most frequently what is lost is cell cycle regulatory genes or growth signaling genes. And so it seems like those are two pathways to get to a more aggressive tumor. And we're modeling all this now in mice. So it's relatively straightforward to start modeling these things. Okay, so that's a really brief tour. I wanted to stick to my time this morning. I do want to just show the members of our lab who contribute a lot. And I've mentioned the work of a few other people over the course of this very brief discussion this morning, showing our funding here and some of our collaborators. And this is really kind of what's going on today. We've had a variety of collaborators over the years, but we're not showing all of them in previous funding and whatnot. But, and again, you can see the EHE Foundation here has been a big funder of our laboratory, and I wanted to expressly thank them for funding this work. They saw that the work had promise at a very early stage, and we've been all in together. It's been a really, really fun ride because I think the foundation supported us. We supported the foundation, and things have gone really well. All right, so now I'm going to transition to the rest of the session, and I'm going to stick around and kind of be involved helping David out with the moderating. But it looks like our first talk is going to be from Freek van Eeden in Sheffield. I don't know, David, are you going to introduce him?
Yes, and just, the morning sessions in general here are going to focus on model systems. And I think it's important to note that, these model systems are critical for both understanding the disease, as well as providing systems to identify targets for therapeutics and for testing therapeutics. And just a reminder to everyone here that it wasn't that long ago where we had no models at all. And it's the existence of these models that's really driven the remarkable progress over the last few years in this field. And so, yeah, we're going to start off the session with Dr. van Eeden.
Okay, sorry. Let's see whether this works. Okay, can everybody see my screen? I hope so. Before I start, I would like to thank the organizers for allowing me to speak here, even though our progress in making a model has could have been better, but you'll see, I think there's light at the end of the tunnel. I need to, Okay, so first, a very short primer on EHE, just that everybody's on the same page. So it's Epithelioid Hemangioendothelioma, which is a rare vascular sarcoma. And in the great majority of cases, it's caused by a translocation of the N-terminal exons of the TAS gene with the C-terminal exons of the CAMTA gene. And this leads to a fusion protein, and it's thought that the oncogenic activity is mainly due to the dominant activation of TAS, which becomes permanently localized in the nucleus. And as Brian Rubin told you, there have been some recent really beautiful models. Sorry, I have to move this down. Some really beautiful models have become available in the mouse. And you might ask, is it still worthwhile, trying to get a zebrafish model? And we think it is actually, because there are a number of strengths of this model, if we're able to make it, that would be really useful to study EHE. First of all, there's strength in numbers. So you can get a lot of material out of a zebrafish. If you create a model, a larval model, the fish will lay 100 to 300 eggs per week. So there's lots of material to work with. The eggs are extremely accessible, and I will come back to this. Also, if you want to combine, an EHE model with loss of function analysis of other genes, this is now really easily done using CRISPR-Cas or CRISPR technology. And finally, of course, it's a vertebrate. So the anatomy of the fish, although there's quite a bit of evolution between a human and a fish, the anatomy can be pretty well related to human anatomy. So we can really, compare the two quite easily. But the most important asset of the zebrafish is its embryo. So it develops really rapidly from a one-cell stage. Within 24 hours, you will get a basic body plan. And within 5 days, you will have a complete larva that is capable of taking care of itself. It can eat, it can feed, it can do everything it needs to survive. Importantly, the embryos are not dependent on circulation, at least for the first 4 or 5 days of development. So even if an EHE model will cause serious defects in blood vessel formation, for instance, that's not a death sentence to the model. You can still use it for quite some time. A really important asset of the zebrafish is its transparency. So there are really useful transgenic lines that very nicely highlight the blood vessels in the zebrafish. This is an example of a fly 1A GFP line where you can really nicely see these intersegmental vessels. And you can actually see these vessels forming live in a living embryo. You can see an example over here where you have, and this is actually a calcium reporter. I'm going to try to start a video here. See whether it works. Yeah. So this is a calcium reporter where you can see live calcium signaling occurring in endothelial cells while they're forming such a intersegmental vessel. And you can see an amazing amount of detail with phylopodia forming. You can see cells dividing. So you have a whole load of readouts that you can use to characterize what an EHE or a TAS-CAMTA fusion might be doing to the behavior of these cells. And importantly, the embryos are really small and they fit within a 96 well plate and they can live in there for quite a few days quite happily. So if you have a larval model, you can use it for in vivo tests of drugs, or if you have a set of candidates, you have 13 different candidates of a drug and you might say which one is the best. Then you can really easily expose these fish and do a variety of readouts to figure out whether they're responding to the drug in the right way. But of course, this is all nice talk and we first need a model. So this is where we've been starting quite some time ago. And the first thing that we did was simply to express a TAS-CAMTA in a zebrafish larva. And this is shown over here where you can see we got these constructs actually from Xuang Ma and Brian Rubin, an inactive construct over here and an active TAS-CAMTA1 construct. And indeed, if you look at the blood vessels in these animals, you can see that the intersegmental vessels are strongly reduced in the embryo. So that was a good sign. Also, we got information about an antibody that might work. And this is a human anti-CAMTA1 antibody, which nicely shows these bright green dots here in the injected embryo. And this shows that it's actually nuclearly expressed. So the protein is also in the right place. But an issue is that, this is transient expression. What we really want is endothelial expression. So we needed a transgene. And for that, we chose to use the Fly1A promoter because this had been the workhorse of endothelial expression in the zebrafish. And it has been very well characterized. So we made a variety of direct and indirect expression constructs, either where the Fly promoter drives TAS-CAMTA immediately, or where we have a step in between where Fly1A drives a GAL4-like protein that then activates a UAS promoter. And behind that, we cloned a whole variety of TAS-CAMTA1 variants. We injected those into eggs, and this is where the frustration came along, because whatever we tried to do, the RNA was not expressed in the endothelial cells. And I'm really summarizing 2.5 years of work, and it was really frustrating. Everything that we did failed. And I can show an example over here. So this is the same GAL4 driver, Fly1A, which drives a UAS GFP. And you can see in the blue stripes over here that there is nice endothelial expression. However, if you use the same driver to use TAS-CAMTA on TAS-CAMTA over here, we only got neural expression. So we were really frustrated and almost ready to throw in the towel. But then we talked to Valerie Kouskoff, and she said, actually, well, maybe Tascamta is interfering with the Fly1A promoter. So basically, you know, you're blocking your own expression. And we decided to follow this up. So now we're actually at the situation where we're going to use the ubiquitin promoter, and we want to control this using CRE. And this is a kind of where we are at the moment. So we've made this is our favorite construct. Here the ubiquitin promoter drives an NLSM cherry between lock sites, and it has a transcriptional stop over here. And we will use this to make the transgenes. And you can see an injection over here where we've injected this construct into zebrafish, and you can see the red nuclei popping up over here. However, if you co-inject this construct with a CRE RNA, you will lock out this bit over here, then ubiquitin will start driving the Tascamta protein. And Tascamta protein is linked up via a self-cleaving peptide to a GFP version. And so if you then co-inject the CRE mRNA, you will start seeing Tascamta expression by means of the GFP neon. And you can see this over here. So we're now seeing light at the end of the tunnel, and this is basically where we are at the moment. But we're growing up the fish that have been injected. We will be able to identify the fish by M-cherry expression, and we're also generating a temporally controllable CRE-ERT2 line where we can express CRE-ERT2 in the endothelial cells to specifically activate the Tascamta in the endothelial cells. We're also, since we're getting a bit paranoid about this promoter issue, we're also looking at other promoters. So we've now a construct where the zebrafish TAS promoter is driving expression of M-cherry and the Tascamta GFP. And finally, we're doing a bit of work to figure out, you know, how Tascamta is able to interfere so badly with this Fly1A promoter, because this is still a big mystery to us. This promoter has been used by hundreds of labs around the world as a endothelial driver, and we don't understand how this can be blocked. And this is all I wanted to talk about today. I just want to acknowledge a few people, especially Eleanor Markham. She's the MPhil student that has been working on this for 2.5 years, and it has been quite frustrating at times. So she's really, I have to commend her tenacity in this. I also would like to thank these people for sharing materials and information. And finally, I would like to thank you, Leonard, and Guy Weinberg for funding and also for mental support throughout a few frustrating years. But I think things are really looking up now, and I hope that everything is going to be, that we will have a model soon. And I'm going to stop here.
Thank you much, Dr. Van Eden. Our next speaker is going to be Dr. Vanoli.
I'm sorry, I'm just having an issue with the sharing. Give me 1 second. Can you see my screen? So good. Can you see my screen then? Sorry.
Yes.
Okay, thank you. So good morning, everyone. So my name is Fabio Vanoli. I work in the Dr. Antonescu lab at Memorial Sloan Kettering, and today I'm going to talk to you about CRISPR-Cas9 model of WWTR1 CAMTA1 fusions. So as already been told, EHE is a rare vascular sarcoma with a prevalence of 1 per 10 to the 6 person. One of the hallmark of EHE is the fusion of WWTR1 CAMTA that is present in roughly 90% of EHE cases, 45 of which have no other genetic alteration. TAS is the protein coded by WWTR1 gene, is a coactivator of gene transcriptions, and is fused to the DNA binding domain of CAMTA1 to generate the chimeric transcription factor. The remaining EHE cases harbor the YAP1 TFE3 translocations. So, as already been mentioned before, one of the reason why the EHE, like other rare sarcoma, are difficult to study is the lack of in vivo and in vitro model. Because of that, the aim of our work is the generation of faithful in vitro model, in our case, cell line of EHE tumor. So before going into the detail of the strategy that we are using to generate this model, so in collaboration with Frank T. Rod in Lyon, we did an RNA-seq analysis from paraffin-embedded tissue of EHE. And the first thing is that in the case, you can see here, EHE clusters separately from other sarcoma types. From these RNA-seq analysis, we extracted genes that are differentially regulated. Here, I'm just presenting 2 genes that are highly upregulated. And out of this list of genes upregulated, we selected 4 genes that we call it the EHE core gene expression signature that we confirm by qPCR in additional 4 EHE tumors from frozen tissue available in the lab. As you can see, the genes, the 4 genes are in the black bar in the EHE tumor up regulated compared to normal tissue and other sarcoma types, clear cell sarcoma in this specific case. So this gene expression signature, this EHE gene expression signature is important for us because that is are the genes that we will use it to validate our model. Now, jumping into the strategy that we are using. So with the development of genome editing techniques, first Zinfinger, then Talen, and more recently the CRISPR-Cas9, modeling chromosomal translocation became relatively easy. And the strategy is based on the induction of 2 double-strand breaks in the 2 loci participating in the translocation, and then on the formation of the fusion translocation by a DNA repair mechanism called non-homologous end joining. Now, this mechanism is what happened in human cells, in vivo and in vitro also, and has only one problem, that is the frequency of the event is relatively low. And so that could make it more complicated to select for clones carrying the translocation. Because of that, during my postdoctoral training with Dr. Jason at Memorial Sloan Kettering, we developed a system where we induce 2 breaks in the participating loci, but instead of relying on non-homologous end joining, we use an alternative DNA repair pathway that is HDR, homology directed repair, in which we provide a template with an hygromycin selectable marker that, if inserted correctly between the 2 ends that we generated, then can give a resistance to hygromycin. In this case, we can select the clones harboring the chromosomal translocations. I'm specifically saying chromosomal translocation, not diffusion, because diffusion at this step is not induced, and we can induce the expression by Cre recombinase, as is shown in this cartoon here. This strategy has 3 main advantages. Number 1, we can select the clones. Number 2, we can induce fusion expression. And the last one is that the fusion is expressed under the control of the endogenous promoter, in this case, WWTR1. So here is just as a proof of principle, is just an example of one of the fusion that we model. This is the EWSR1, WT1 fusions. So as you can see in the clone here, the fusion expression by RT-PCR is achieved only in cells after Cre expression. And this is true at the RNA level, and also, as you can see here, at the protein level. And we model it in different fusion types. So now when modeling any cancer-relevant mutation, including also the translocation, one of the important points is to choose the right cell context. So to give us more flexibility, we use an approach that starts modeling the chromosomal translocation in human embryonic stem cells. And we use human embryonic stem cells because they are relatively easy to use. They can be isolated as single clones and can be passed almost indefinitely. So once we generated the chromosomal translocation in the human embryonic stem cells, then we differentiate to any cell line using the pluripotence capacity of the human embryonic stem cells. And of course, at that point, we can express the fusion and study the effect of the fusion in the different cellular contexts. This system gives us more flexibility, number one, and allow also to, in the case of sarcoma, for example, where the cell of origin is not clear, to consider different cell lines. And we successfully model in a recent publication 2 different sarcoma types. Of course, in the case of EHE, the cell of origin is well known. And so what we will do, it will be to differentiate mainly this, our human embryonic stem cells to endothelial progenitors. So now here, I summarize the WWTR1 CAMTA fusion isoform that has been identified in EHE by our lab and by other groups. So here are listed the publication. And today, for this presentation, I'm focusing on the isoform, the fuse exon 4 from WWTR1 with exon 9 from CAMTA1. And here is the strategy that we use it. So as you can see, we induce 2 breaks, one in the intron 4 of WWTR1 and one in the intron 8 of CAMTA1. And then we provide a donor template with hygromycin. And then we select clones resistant to hygromycin are then screened by PCR. And we have a 3 PCR. We have a 5' PCR and a 3' PCR to be sure that the donor template is being corrected in the right loci, and then what we call it an out-out PCR across the translocation. As you can see here, in the 3 representative clones number 1 is positive for 5' PCR, 3' PCR, and also out-out PCR. So basically, we carry on this clone, and the first thing that we did it was to remove hygromycin by Cre-induction. So now, experience from different fusion demonstrate that the expression of the fusion can be toxic for the cells. And so basically the cells can die. So we can monitor if the cells can sustain the presence of the expression of the fusion at the genomic DNA level, comparing the population retaining the hygromycin marker and the population without the hygromycin marker. As you can see in this time course experiment, the ratio between the hygromycin populations and the population expressing the fusion remain constant throughout the experiment up to 26 days, suggesting that it is indeed not toxic. Of course, we are interested to see what about the fusion if it is expressed. And what we found out is that the express, you can barely see the product. So basically, it looks like the fusion in this cellular context, in the human embryonic stem cell, is not well expressed. So then we wonder if it's really just a problem with the cell line, and because of that, we differentiated the cells to human embryonic mesenchymal progenitor. And we use this one because it's a cell line that we can easily differentiate in the lab, and we have already experience with other sarcoma types. And as you can see, indeed, when we express the fusion in mesenchymal progenitor, we can see a high-level transcript. So now, as I mentioned before, we are interested in understanding if we are interested in seeing the expression in endothelial cells. So we differentiating using a commercially available protocol our human embryonic stem cells with the WWTR1 translocation to endothelial progenitors. We confirm that in the 2 markers, 2 endothelial markers are positive, CD34 and CD31 are positive in our cell line, and then we express the fusion. And once again, also in this case, those are 2 different cDNA concentrations. You can see that the expression of the fusion is much higher in endothelial cells, like observed in the mesenchymal, compared to human embryonic stem cells. So as I mentioned at the beginning, so we extracted from the RNA-seq analysis of EHE tumor, we extracted the core gene signature. For what we call it the core gene signature, 4 genes that are highly upregulated. And to validate our cell line, we check if indeed in those cells expressing the fusion, also those genes are upregulated. Of the 4 genes, only 1 is upregulated. So we wonder if, once again, the endothelial cells, the progenitor, the endothelial progenitors that we are using are indeed the right systems. So because of that, in collaboration with Lai-Teng Ang at Stanford University, we decided to further differentiate these endothelial progenitors, the one that we think we have it. We committed to a more to a further step in the differentiation process to artery and vein. So we are in the process of doing this experiment, but in the meanwhile, we compare the gene expression analysis of artery and vein cell line provided by Ang Lab, derived from iPSC with the RNA-seq expression of our EHE. As you can see here and here, 95% of artery and vein genes are shared with the EHE tumor, with analysis of EHE tumor, suggesting that it's probably worth considering modeling our fusion or expressing our fusion also in these cell types. Of course, the general idea would be that it would be nice to study different level, the expression of the fusion at different level during the differentiations. So in conclusion, we define a core gene signature for a EHE tumor. We select and we isolate clones harboring the WWTR1 CAMTA translocation in human ES cells. We induce the gene fusion in human ES, and that results in a low transcript level. And as a consequence, we believe that's why we don't see any alteration in cells proliferation. And that is opposite to what observed with the different tumor types, sarcoma types. When we differentiate the human ES to mesenchymal or the endothelial, on the opposite, we see a higher expression. And the expression of WWTR1 CAMTA in the endothelial progenitor only partially recapitulate the EHE core gene signature. So there's a future direction. What we are doing now is that we want to model the different fusion variants for the WWTR1 CAMTA and also YAP1 TFE3 in human ES cells. We will differentiate the cells to artery, vein. We will repeat also for the different isoform also in the endothelial progenitor. So to make the system more flexible, we are also targeting our cell line with a TET-on or with a CRISPR inducible system to achieve higher expression of the gene fusions. And then we will characterize our cell line to determine transformation capacity. And the final goal would be using our cell line for drug screening and hopefully to develop also the xenograft mouse model for EHE. And with that, I want to thank, of course, Christina to give me the opportunity to present, to work on this project. The lab member, present and former lab member, our collaborator at MSK, Built Up, Sam Singer, Ping Chi, Frank Tirod, Lyon for the EHE RNA-seq analysis, Lai-Teng Ang at Stanford for the endothelial differentiation and the RNA-seq, the funding, and of course, the organizer for giving me the opportunity to present. Thank you.
All right, our next speaker is going to be Dr. Kouskoff. And just a reminder for attendees, if you do have questions, type them.
Okay, so can you hear me?
Yes, we can.
Okay, perfect. So I want to thank first the organizer for giving me the opportunity to present our data. So today I'm going to talk about our study on the consequence of TAS-CAMTA1 expression in primary endothelial cell. And all the work I will be presenting was performed by Emily Neal, who is a very talented PhD student in my lab, and she received a PhD fellowship from the EHE charity in the UK. So in our lab, we use embryonic stem cells. So it's a very good transition from Fabio's talk. So we use embryonic stem cell that can be differentiated to various lineage, and we differentiate those embryonic stem cells to endothelial progenitor. And those endothelial progenitor are sorted from the bulk culture and culture into conditions that allow the endothelial to further mature. And at various time points, we can induce the expression of TASCAM-TA1 using a doxycycline-inducible system. And then we can monitor TASCAM expression by the flag tag or by GFP or as doxycycline-induced expression of both TASCAM, TA1, and GFP. Okay. So one of the questions we try to answer is how does TASCAM-TA1 alter the transcriptional landscape of endothelial cell? And the beauty of the system that we use with the ESL is that we can monitor the onset of TASCAM expression, and we can produce enough cell to do a lot of analysis. So in this case, to look at the landscape, a transcriptional landscape of endothelial cell, we induce the expression of TASCAM-TA1 for 24 hours, and then we harvest the cell, purify the population by flow cytometry, and perform high throughput RNA-seq. So when we induce the expression of TASCAM-TA1, we can distinguish two types of population that are GFPI and express high level of TASCAM-TA1, and population with intermediate level of GFP and express low level of TASCAM-TA1, and cells that are negative for GFP. So we sort those three populations, and in parallel, we also perform the RNA-seq on cells that were not induced with the doxycycline. So we have 4 populations that we tested for RNA-seq expression. So what we observe is a very good correlation between our level of expression of GFP and the RNA expression for TASCAM-TA1 with the 4 groups that fall nicely onto this level, decreasing level of expression. But what was really striking is that we saw that in the TASCAM-TA1I cell population, we saw a huge amount of genes that were dysregulated. It's about 4,500 genes that were either downregulated or upregulated. And you can see on this heat map that about half of the genes were downregulated while half of the genes were upregulated. We saw much fewer genes being upregulated in the cell that express low level of TASCAM-TA1, about 500 gene differentially regulated. So when we look closer at the signature of those genes using gene set enrichment analysis, we saw that both the HIPPO pathway and the YAP and TAS cancer signature were highly enriched. In the TASCAM-TA1I expressor population, we didn't see so much change in the TASCAM-TA1 low population. But what was really interesting is we took the gene set that was defined by Seavey and collaborator in the lab of Brian Rubin, and they've put a list of genes that define human EHE. And what we see in our cell that express high level of TASCAM-TA1 is that they are highly enriched for this EHE gene set. So it's very important because it's telling us that we are looking at cells that are closely resemble the human EHE cells. So in summary of this first part, what we saw is that within 24 hours of TASCAM-TA1 expression in endothelial cell, we see a massive change in the transcriptional landscape of those cells with notable upregulation of the TAS signature and of the EHE signature. And I didn't show that, but downregulation of the endothelial identity. So we also were very interested in looking at how TASCAM-TA1 expression altered the biology of the endothelial cell. And one of the first things we observe is that when you induce the expression of TASCAM-TA1, you block the cell in the S phase of the cell cycle. So this was done by EDU incorporation. And what you see is that there is a decrease in the frequency of cells that are in the G10 stage of the cell cycle, accumulation of cells in the S phase, but those cells do not transit to the G2M. So those cells are arrested in the S phase of the cell cycle. And there are many reasons why cells might be arrested here. But one of the reasons is if they accumulate DNA damage. So we started looking at DNA damage. And one way to look at it is to stain for the phosphorylation of the histone H2AX. And histone H2AX mark double strand break on the DNA. So the untreated cells don't show much staining or positive control of DNA damage H2O2 to show increased activity of the gamma H2AX. And when the endothelial cells are treated for 24 hours with doxycycline and start to express TASCAMT1, and we can monitor that with the flag expression, we also see a huge accumulation of gamma H2AX in most of the cells that express the flag. And this is the quantification of this. And you can see that it's very significant. So to make sure that what we're looking at are DNA double strand break, we perform a COMET assay. And in this assay, single cells are deposited on gel and they're migrated. And if the DNA is undamaged, it stayed in the head of the COMET. But if the DNA is damaged, forming double strand break, it will migrate as a tail. And you can see that the untreated were very few tail DNA. But in the cell that express TASCAMT1 or the cell that are treated with H2O2, we see an increase in DNA tail, and this is again quantified here. And this demonstrates that TASCAMT1 expression lead to double strand DNA break. So for a cell, there is various ways of repairing DNA double strand break, but in the space, typically the accurate DNA repair occurs through the homologous recombination pathway. And this pathway involves the recruitment of BRCA1, which is become phosphorylated and will be recruited to the double strand break and help with the deposition of RAD51 protein that protect double strand single cell DNA break. So we looked at the recruitment of BRCA1 in the cell that were allowed to express TASCAMT1 for 24 hours. And what we observed is there was no recruitment of BRCA1, unlike the control cell in which treated with H2O2, in which you can see the accumulation of BRCA1 at the foci of gamma H2AX staining. And similarly, we didn't see any accumulation of RAD51, while those were clearly visible when the cells were treated with the H2O2. So what we are seeing here is cells that start to express TASCAMT1 within 24 hours. This induces a lot of DNA break, but the homologous recombination DNA repair pathway doesn't seem to be activated. So there is two possibilities when the double strand DNA break are not repaired. Either the cell undergoes senescence, and generally this is a tumor suppressive process, but it might lead to acongenicity if the senescent cell accumulate. But the other alternative leads to genomic instability, because the DNA will be repaired but using an alternative pathway, which is error prone, and this is a hallmark of cancer. So first we look at the accumulation of senescent cells, and this they can be visualized by the expression of P16, which is a senescence marker. And you can see that 24 hour for expression of TASCAMT1 leads to the accumulation of cells that express P16 and the flag, which mark TASCAMT1 expressing cell. You see very few of those cells in the control population. And also what we observe is that if we keep the culture going for a long time, here it's 3 weeks, what we start to see is that in the presence of doxycycline, so TASCAM is maintained, we start to see the accumulation of those cluster of cells that express very high level of GFP. So we are currently looking at those cluster of cells to see if they have accumulated, if they have additional mutation. We never detect those cluster of cells in the controlled culture. So in a summary, in a take-home message, what we have been studying at the onset of TASCAM expression is that as soon as TASCAM become expressed, within 24 hours, we see an accumulation of gamma H2X phosphorylation and mark, an accumulation of double-strand DNA break. This results in arrest in the S phase of the cell cycle, and the DNA damage response is not activated. We are currently trying to understand why this DNA damage response is impaired, but what we see is that no repair will lead to senescence, so we see a decrease in proliferation and an increase in P16 expression. But what we think is happening if the DNA is repaired by the error-prone DNA pathway, DNA damage pathway, then secondary mutation will occur in those cells. They will re-enter the cell cycle, the proliferation will increase, and this may lead to aggressive EHE. And as Brian was showing earlier, about half of the EHE have secondary mutation, and a lot of those mutations are in cell cycle and in DNA damage response. So it does fit our hypothesis. So just to finish, I want to thank the organizer for giving me the opportunity to present the work. So as I said earlier, Emily Neal did all the work with the funding from the EHE Rare Cancer Charity UK. Yeah, thank you for listening.
And thank you to all of our presenters. We've got a little time for some group Q&A here. And we've got a few questions that have come in from the group. And I guess the first question is really, I think, directed to anyone on the team here. The question is, what is the artery vein specificity of clinical EHE features? Any volunteers to take that one on?
Well, I may be the only person who sort of looks at EHEs under a microscope. So there's no specificity for artery vein. In fact, you can't. Occasionally you see an EHE arising in a larger blood vessel that's identifiable, both arteries and veins, but typically you do not. And so there's no specificity. I'm not sure exactly how to answer that question.
All right, our next question is for Dr. Vanoli, and it's a question related to your model system and getting to the issue of if you think any of the effects you're seeing are based on the context of the culture system as opposed to the differentiation state of the cell.
It is possible. It is possible. We are still trying to understand what's the issue. The only thing is, you know, we have, if I understood the question, so we have proof with a different fusion that the expression is really high, starting in human, yes. And you know, we are not really sure. There is also one aspect that I didn't mention for brevity, but it looks like that the promoter of WWTR1 is expressed, is well expressed in the human, yes. It's just when is when it's combined with the CAMTA1 that we see a lower expression, and that is what we still do not understand, because the hygromycin gene is expressed. Indeed, the cells are resistant. So it could be a combination of things anyway.
Thank you. And we've got a question here about the Fly1A promoter, and the question is, can you introduce TEAD binding sites into the promoter in anticipation of boosting the TAS-CAMTA1 expression?
We could do that. I think that's a very interesting idea, and it's something where, you know, it might not be too difficult to, you know, now with gene synthesis to put a few in there. So this is something that we maybe should follow up and see if we can get some auto-regulation going that basically activate your own construct. So that's a very good idea. We'll think about that, whether we can do that.
And one more question about that system. Have you confirmed that the repression of the Fly1 by TAS-CAMTA1 by RNA in situ measurement?
So we haven't. So our main aim was to get a model, and we are looking in what is going on. So we're basically trying to split up the TAS-CAMTA protein and see whether there are any bits of it that specifically act on the promoter. And so far, things have been rather confusing because the TAS does not seem to influence the fly promoter, but any bit of the CAMTA that we have taken seems to be very, and this is in transient experiments, so it's a bit difficult to interpret, but any bit of the CAMTA seems to be very negative, affecting the expression. So the thing is, we want to get a model. We don't, and initially at least, we're not that interested in it. So we've been focusing our work on getting an EHE model in a fish rather than figuring out why the previous thing went wrong. Although there is, it is something where it keeps us awake at night, or at least it keeps me awake at night. Because we're still thinking, is there something else that we missed that is not right? But so far, all the controls seem to be behaving, and it's really, there's something odd about the fly promoter, we think.
Excellent, and that hit the initial batch of questions that have come in. Again, thank everyone here on the group, and we've got we're a little bit ahead of or behind time, so we'll catch up here by moving on to the next session, which is a video by Dr. Barker.
Hi, I'm Holly Barker from the EHE in Melbourne, Australia. I'm going to tell you today about the Stafford Fox Rare Cancer Program and describe how we are using genomics and preclinical models to find better treatments for rare cancer patients. And so I'm sure most of you are aware that rare cancers are those with an incidence of less than 6 cases per 100,000 people each year. And super rare cancers are those with less than 0.1 cases per 100,000 people. And this is where EHE is. However, if you put all rare cancers together, they actually account for 1/5 of all cancer diagnoses and almost 1/3 of all cancer deaths. So there's a big unmet need here to study these cancers. Outcomes are inferior to those for more common cancers, and this is because of a lack of preclinical models and evidence-based treatment data. And this is where our rare cancer program comes in to develop these models to find better treatments. We focus on gynecological cancers initially because 50% of gynecological cancers are classified as rare. So I'm just going to run you through the program and then describe some of the EHE cases we have within our program, and then end with an example of how we're using our preclinical models for personalised medicine in other rare cancers. So the EHE Stafford Fox Rare Cancer Program is a national program in Australia, and we're aiming to build a national rare cancer resource to underpin rare cancer research. And you can see the sites where people can participate in the program around Australia. We established this program at the end of 2016, and it has a remote consent option. We collect clinical data and tissue, and we've developed a number of research projects for specific rare cancers. There are a number of recruitment methods through the ARC portal, direct referral, and Cartwheel, which is an online consumer-led platform for patients to enter their own clinical data. And the PIs for the program are Professor Claire Scott and Tony Pappenfuss, and we have a large team of people processing the samples and developing the research projects. To date, we have consented almost 500 patients to the program with 590 tumours, and these cover 130 different subtypes of rare cancer. So you can see many of these cancers are rare gynecological cancers, as I explained, and we're accruing quite large numbers of some of these subtypes of rare cancers. And we've also consented 8 patients with EHE, so I will discuss these a little bit further. So this is quite a busy slide, but it shows you what happens when a patient consents to the program. We will characterise their tumour by immunohistochemistry and sequencing. You can see in this red box up here. And this table shows the different types of sequencing we'll do and the numbers of cases so far we've completed this sequencing. If we get fresh tissue, we'll also try and generate in vivo preclinical models, PDX models. And to date, we have generated 68 different models for rare gynecological cancers, 2 non-rare gynecological cancer models, and 2 non-gyne rare cancer PDX models. We also try and develop cell lines and organoids, and these are for in vitro, more rapid assessment of drugs in vitro. And hopefully we'll be able to find new treatment options for these rare cancer patients. So as I mentioned, we so far have 8 EHE cases in the Stafford Fox Rare Cancer Program. And you can see here the sorts of analysis we've done on these cases so far. So they are positive for a number of endothelial markers and other markers used to diagnose EHE. And we've been able to complete some fusion analysis to identify 3 of these cases with the common WWTR1 CAMPTA1 fusion. And we identified 1 case with the more rare YAP1 TFE3 fusion by whole genome sequencing. And we're currently awaiting for the completion of some other molecular analyses. So here you can see the results that we get from the TrueSight fusion panel. Interestingly, case 1154 and 1455 have the exact same breakpoint in this fusion gene, with the first 3 exons of WWTR1 fused to the last 16 exons of CAMPTA1. Case 1368 has almost the similar exons and fusion with a slightly different breakpoint to the other 2 cases. Case 1130 actually had whole exome sequencing prior to consenting to our program, so this was on an early needle biopsy. However, due to low tumour purity, this was relatively unsuccessful. They only looked for the more common fusion, and that was not detected, and there are just a couple of mutations of unknown clinical significance identified. Once they consented to the program, we were able to obtain a fresh sample from surgery, and this was indicated to have 30 to 35% tumour purity, and so it was able to go for whole genome sequencing. And you can see on this circus plot that there was a very clean molecular background, and the rare YAP1 TFE3 fusion was identified. So I'd just like to finish by giving an example of another type of rare cancer we're studying in the program. This is uterine leiomyosarcoma. And we have a project where we do molecular analysis of these tumours, and if we identify a mutation that predicts these patients might respond to PARP inhibitor, the patient is given compassionate access from AstraZeneca. We also look at the response by developing preclinical models and use these also to identify other treatment options for these patients. So here's an example of one of our cases, 1122, who had a range of different disease events and a number of different types of therapies, and was actually very unwell when they consented to our program. We carried out whole genome sequencing on the tumour and identified a deletion of BRCA2. So this patient received compassionate access to PARP and had a partial response at 3 months. At the same time, we developed a PDX model, and you can see the validation of the PDX model up here on the left. And we put this on a range of different treatments and different dosages of the PARP inhibitor olaparib. And you can see the highest dose showing some response in green here. Interestingly, this model also had a really good response to cisplatin, and ULMS patients would never normally be given cisplatin in the clinic. So we thought, what if we combine cisplatin with a PARP inhibitor, which is okay for patients to receive, and you can see in the purple line here we had an even better response. And the patient was actually receiving this double combination in the clinic as well after they progressed on PARP alone, and they had a partial response at 3 months and no tumour activity was seen at 6 months. So this is a great example of how we're using our PDX models to validate what we found molecularly and also identify additional treatment options for different rare cancers. So I'd just like to finish with my big acknowledgement slide. There's a big team of people involved in consenting patients. You can consent through Cartwheel or through Marita Black at BioGrid, and these people involved in processing of samples and development of the projects. And of course, we couldn't do this without the funding from the staff.
All right, we continue on with the second part of our morning session with the next set of speakers, also talking about models. Our next speaker up is Dr. Al-Jazrawe.
Good morning, everyone. All right, so thank you very much for the opportunity to give this talk. And I'll be telling you a little bit about our experience with rare sarcomas in general and how we would love to bring all these elements that we've learned so far into the challenge of model generation for EHEs. As you know, the challenge of generating cell lines for EHE is a very difficult challenge. So I think a shared vision for us is that we want to understand how the molecular profile of a given tumor relates to the list of genes or drug targets that a given tumor would be, that are essential for the growth of that given tumor. And what we call those are genetic or compound dependencies. And so as we profile multiple patients, many patients' tumors, and understand how which drugs selectively kill those tumors in culture and what molecular features these tumors display, we can understand and build a map that we call the cancer dependency map, which is basically a reference of how biomarkers relate to dependency relationships or which sensitivities a given tumor will display. Now, a key component of generating such a map is the use of existing or generating new patient-derived cell models so that we can conduct these genetic or compound library screens. And as you know, a challenge in the EHE field and rare sarcomas in general is the existence and also the ability to generate these cell models. And so our lab tries to focus on this problem. And so one of the biggest problem is the reality that these tumors are rare and they're scattered around the nation. And so the traditional single institute, single lab approach typically means that you get 1 or 2 samples per year to try and generate a model from, which means A, it's difficult to have sufficient samples to understand truly how a given media condition or given supplement regimen leads to success or failure of generating a model. And it also means that it's a very long time to build up this type of knowledge set. And so we've led an effort for a nationwide strategy to collect a patient-led consent program so that we collect patient samples from throughout the United States so that we can increase the number of samples that we can receive in our lab. And that's organized through pattern.org, but also efforts by the EHE Biobank is also similar in this effort to try and increase the number of attempts that we have to generate cell models for EHEs. Now, the other challenge, in addition to lack of patient samples to begin with, is the challenge of what happens in the lab to generate a model. Typically, a traditional path has been to try 1 or 2 media conditions that have either anecdotally been successful in similar sarcomas or similar diseases with similar molecular features. But as we've learned from our experience at the cancer cell line factory at the Broad, that increasing the number of media conditions that you try, increasing the number of attempts that you try, improves your success rate of generating a model. The problem that's associated with increasing the number of attempts is logistically, it's difficult to credential genomically every condition that you're growing. And so typically it also means that you're picking and choosing which conditions you want. And so it's similar challenge to the traditional path of not having a lot of data to support whether a given media condition is doing better than another or a specific supplement is helpful or not. And so what we'd like to ask is how can we scale up this strategy? How can we go from a patient sample, we attempt multiple media conditions with technical replicates so that we can say something about the effect of a given ex vivo condition on the growth of a tumor and on its impact on cellular composition. And so to achieve this, one of the rare sarcomas that I've studied is desmoid tumors, which shares a lot of the similar challenges and opportunities that EHE shares. So I'm going to share some of our progress in that area. And to begin the strategy of trying to credential a lot of media conditions at once, we adapted a barcode sequencing approach so that we can culture a tumor sample, try out almost 100 conditions, but then at the same time, be able to collect DNA from the sample across all conditions, study the presence of mutant allelic frequencies across all conditions, and then demultiplex the results so that we can identify which condition is enriching for the tumor population, which condition is enriching for non-malignant cell growth or not even allowing for any cell growth to happen. And so we applied this, as I mentioned, to desmoid tumors. It's a rare sarcoma caused by beta-catenin mutation. As Dr. Rubin mentioned, in EHEs, about 94% of patients display the WWTR1 CAMTA fusion. In the case of desmoid tumors, it's very similar. In this case, over 90% of patients express a mutation in beta-catenin in one of the phosphorylation sites that are important for its degradation. And so the search space for identifying the mutant allele is very narrow. And so that makes it helpful to conduct these type of multiplexed screening in a quick fashion without having to look through all 20,000 genes to find potential tumor markers. And the second, and as also you can relate to, is the challenge of developing cell lines. So less than 5% of desmoid tumor material that comes into a lab can actually turn into an expandable long-term cell model using traditional methods of conducting cell cultures. And so to pilot this type of strategy, we took a few samples from desmoid tumor cohort. We credentialed their beta-catenin status from the primary tumor. And then those that displayed a beta-catenin mutation, we grew them in a 96-well plate with 32 conditions in triplicates. And using this strategy, we can measure the allelic frequency starting at the first passage. So we need minimal material, unlike a typical traditional DNA sequencing method where you need to pellet a good number of cells so that you can do deep sequencing on it. Here, we only need very limited material as you're passaging the cells. And so you're collecting enough information as you're going from P1 to P2 to P3 and so on. And then you're measuring the amount of in this case, beta-catenin mutant alleles and see how it's changing over time and also across multiple conditions. And as you can see in the top figure here is that as early as the first passage, we can see that some conditions are enriching for the tumor population, and some conditions show that non-malignant fibroblastic growth in this case is enriched. And so that means two things. One is that as early as the first passage, we can essentially focus on few conditions. So we can focus our efforts and resources on the few conditions that show high success probability. And the second thing is that because as early as the first passage or even the second passage, as you can see in this figure in the bottom, that we can start to see trends of which medias and which supplements are improving. So potentially we can think about shifting the perturbational studies that we're planning to do rather than in a very late passage. We can do it as early as P1 or P2. That way, potentially we can study a diversity of patient samples using this method. And so we would love to apply this for EHEs. And it starts with creating a quantifiable readout. Obviously, the fusion is the most direct and best assay that we can go for to identify which conditions are enriching for the fusion expression and which conditions are not. And it also starts with collecting sufficient samples so that we can collect enough information to say something about which supplements and which media conditions are enriching or hurting the tumor composition. So we started, this is just super early work to show sort of the bits and pieces of the process, and we would love to put it all together in the future in a more comprehensive screen. But we started with a sample that was a pleural effusion that was known to be fusion positive. And we did 2 or 3 different strategies. One is to culture the cells and credential them as is. And we also did CD45 depletion to remove a lot of the immune cells that we know come in those pleural effusion samples. And we also did CD31 enrichment. The goal is to enrich for the tumor cells so that we have a better shot at culturing the right cells, and also for mimicking essentially different conditions that can change tumor purity and see if our assay can compare the differences in fusion expression. So thanks to Dr. Rubin's advice, we developed a number of primer sets to look for the fusion, and then subsequently to look for the specific breakpoint using Sanger sequencing. After that, we developed a qPCR probe to use that in a qPCR experiment to compare the relative expression of the WTR1 CAMTA fusion. And as you can see in this figure, we find that the different enrichment methods do indeed increase the relative expression of the fusion, suggesting that we can indeed enrich for more tumor cells when we first start the sample and initiating it in culture, and also that our method can help us quantify the relative expression across different conditions. And so now we're sort of in this period where we're collecting primary tumors, credentialing them, and then putting them in a queue to start this media screening strategy. And this is where we need the community's help with in collecting more samples that are known to be highly likely to have a fusion positive, so that we begin our screening strategy so that we have a lot of replicates to study the impact of different conditions and different supplements. And so as next step, as I mentioned, we wish to include and acquire additional samples and improve our sample selection. As you saw in the previous slide, that only a minority of samples we've been receiving actually have a fusion detected using RT-PCR. We also like to test the feasibility of enriching for CD31 positive population across culture, hoping that that could help enriching for the tumor population. And as we learn more from increasing replicates, we can talk more in the future, hopefully, about which media conditions and which supplements can help establish EHE cell models. And just lastly, as we're collecting this experience across all of rare sarcomas in our lab, we're also thinking about, what about skipping the cell model generation step? As I mentioned, our technique with barcoded sequencing, we can look at the effect of the mutant allelic frequency as early as the first or second passage. And so can we use this feature instead of different media conditions? We can look for different compounds and see how they are impacting tumor purity across time, potentially alleviating the need of producing long-term expandable cell models. We're also using single-cell RNA sequencing to study tumors at their earlier state when they are still mixed with normal and tumor cells. And we're also using morphology because we think the single-cell resolution that morphology gives and the ability at a very high scale to image a lot of cells and then identify their identity and whether they're responding to a drug or not by morphology alone. I think this has a good potential to work in EHE samples that are early, but we cannot generate a cell model for. And with that, I'd like to thank all of our collaborators and members of the Boehm Lab, and also the EHE Foundation, and especially the patients, all the patients that donated their tissues for research. So thank you very much.
Our next speaker in this session is Dr. Seavey.
Good afternoon, morning to everyone. Let me just pull up my slides. Are you able to hear me and see me?
Yes, on both fronts.
Absolutely. So thank you so much for letting me speak today. I'm going to be talking not so much about the previous work that we've published, but actually a fairly new and fairly exciting story that has come out of the previous mouse model that we've developed. As previously mentioned, Caleb Seavey, I'm a research fellow at in Brian Rubin's lab, as well as a clinical resident in the Department of General Surgery at the Cleveland Clinic. And I'm going to be talking about the development of a cell-based transplantable model of epithelioid hemangioendothelioma. So as Dr. Rubin previously mentioned, we've developed a mouse model of EHE where we're able to replace the wild-type WWTR1 allele with a WWTR1 CAMTA1 allele under the control of a Cre recombinase. And when treated with tamoxifen and with a tamoxifen-sensitive Cre recombinase, either a ubiquitously expressed or a endothelial-specific Cre, mice develop tumors that are grossly representative of aggressive type EHE, both developing tumors along the peritoneal surface as well as involvement of multiple visceral organs. As well, these demonstrate they're a histologic mimic of the human disease, and they display the characteristic immunohistochemical staining of EHE. And then finally, they display the characteristic transcriptomic defining features of human EHE. So while this model is very great, and it was able to really demonstrate that TAS-CAMTA1 is sufficient to develop EHE, there's some limitations to this model, specifically the time in which it takes to develop tumors. The average time to develop tumors is somewhere in the realm of 8 to 9 months in this model. As well, there was an incomplete penetrance with this model, and unfortunately, mice only develop signs of disease at a very late stage, such that if we're going to try to do therapeutic testing, it would be very difficult to say when we should start therapy. So with this idea, we wanted to develop a EHE cell line from our GEM model. However, this was ultimately unsuccessful because of fibroblast overgrowth in our conventional EHE model. So I'm going to bring up this paper that was published 2 years ago, where they identified secondary genomic mutations associated with advanced stage epithelioid hemangioendothelioma. And the most common of these was pathogenic variants in CDKN2A. And so with that, we developed a CDKN2A variant EHE model, where we took our CDH5 endothelial specific Cre driver and paired it with our conditional WWTR1 CAMTA1 fusion system, and then finally introduced another mutation, which is a conditional CDKN2A knockout. And so these mice develop tumors that are histologically representative of both the parental, the wild type CDKN2A mice, as well as human EHE, and they display the characteristic immunohistochemical staining for seen in both human EHE and our previous mouse model. And so we wanted to see if we could generate cell lines out of this new line. So these are 3 representative tumors that we performed mechanical dissociation and followed by ex vivo expansion. And so 2 of these tumors have a biallelic loss of CDKN2A, and then 1 of them has a monoallelic loss of CDKN2A. And so sure enough, they were able to expand them ex vivo. These are representative morphologies of these cell cultures, and you can see that 3.6 displays this spindeloid morphology, whereas 3.17 and 3.26 display more of this epithelioid cobblestone pattern. Interestingly enough, all of these morphologies are characteristic of endothelial cells in culture. And so with this, we wanted to further validate that we actually have developed an EHE cell line. And so the first question is whether or not these express the TC oncoprotein and the WTR1 CAMTA1 transcript. And so this is just a representative of one of our cell lines. This has been true for all 3 cell lines. So MS1 is an endothelial cell line of pancreatic microvascular cells, and obviously they do not express TC. whereas our cell lines were able to identify TC in both using multiple primers, and our murine EHE tumors also are able to express WC. Similarly, we were able to identify this at the protein level using an anti-CANTA1 antibody. Next, we want to demonstrate that our cell line is able to recapitulate the transcriptional profile of human EHE and murine EHE. So we had previously demonstrated that human EHE is significantly enriched in YAPTAZ target genes, and similar to our previous findings, our murine EHE cell lines are similarly enriched in these targets. We then wanted to identify whether or not our EHE defining gene set, which was generated from cross-tumoral comparison of multiple endothelial tumors as well as liver, and we were able to generate this 93 gene set. And this 93 gene set that was highly enriched in both human EHE as well as murine EHE is significantly enriched in our cell lines. And then finally, we had previously done single-cell RNA sequencing on murine EHE tumors, and we were able to do cross comparison between tumor-resident endothelial cells as well as tumor cells, both that are actively dividing as well as cells that are not actively dividing. And we generated a gene set that was overrepresented in our EHE cell population. And this was significantly enriched in our murine EHE cell lines. So because we're able to demonstrate all of this work in vitro, we wanted to demonstrate that we were able to actually generate tumors in vivo. So we performed subcutaneous injections of two of our cell lines, 3.17 and 3.6. And 3.17 is able to generate these large, bulky tumors in the subcutaneous space that are histologically identical to the human and murine EHE and are characterized by the presence of mixed with hyaline stroma and vacuolization, as well as infrequently in the 3.17 group, we're able to develop spontaneous metastasis to the lung and to the liver. And interestingly enough, one of our other cell lines, 3.6, while they develop these small diminutive tumors in the subcutaneous space that are histologically identical to human EHE, they universally develop metastasis to the lungs and to the livers, and you can see the frequency of metastasis in this plot right here. The time to development of tumors, or excuse me, to the development of end stage, which we call death with tumor morbidity, and a tumor size greater than 1 centimeter, is virtually identical between our two cell lines. And we can see the tumor growth of 3.17 tumors here. As well, because the primary location in which we identify our murine EHE gem model is intraperitoneal, we performed an orthotopic injection of 3.17, and we're able to demonstrate that they display significant invasion of the entire peritoneal space, with obliteration of the liver-kidney surface, complete involvement of the pancreas, and involvement of the diaphragm. And similarly, these are defined by the histologic phenotype seen in human EHE. And then finally, we perform intravenous injections at 3.17, and we're able to demonstrate the development of metastatic lesions within the lungs upon intravenous injection. Abilities to develop tumors, but I think we have been able to make great strides in developing a EHE cell-based model where we're able to actually test therapeutics. So I'd like to thank my lab as well as our Robert E. Peterson Foundation, as well as the EHE Foundations and the Kovach Foundation. With that, I'd like That's about it.
Our last speaker in this group is Dr. Zaffaroni.
Morning, everybody. In our institution, we are currently developing preclinical models directly derived from the patients of rare and very rare soft tissue sarcomas. To generate these models, we obtain from the surgeon tumor specimens that are analyzed by the sarcoma pathologist. They provide us with small fragments that are injected subcutaneously into the flanks of SCID mice. When the tumor starts to grow, it is removed from the mice and retransplanted for several passages in SCID mice. When the tumor reaches passage number 3, we consider the PDX established. At this point, the PDX undergo an histological examination and a molecular characterization to demonstrate the fidelity with the originating clinical tumor. Whenever possible, we try to obtain also cell cultures from the PDX in terms of 2D and 3D cell cultures that are very useful for drug screening purposes and also for carry out functional assay. Thus far, we have a library of 28 different PDX that we generated for different soft tissue sarcomas histologies, including very rare histologies. The percentage of success in establishing the PDX is different in the different histologies. And in more detail, for the case of Epithelioid Hemangioendothelioma, we started to transplant biopsy, but we were not able to generate PDX from them. And then we moved to surgical samples. Thus far, we implanted surgical samples from 4 different patients. In one case, we were able to establish the PDX. There is another model ongoing, and in the other 2 cases, we failed to do that since the tumor stopped to grow at the second passage in the animal. First of all, we look for the consistency between the PDX and the originating clinical tumors, and we did that in terms of histomorphological analysis, the presence of the fusion gene, and then also by comparing the overall transcriptomic profile as detected by RNA sequencing in the patient sample and in the PDX. And you can see that the profile is almost superimposable as demonstrated by the regression coefficient of 0.92. We were also able to establish a cell line from the PDX. First of all, we assess the presence of the fusion in the cell lines, and then we were able to grow this line as 2D monolayer culture and also as 3D spheroids culture. We recently injected these cells into the peritoneum of SCID mice in order to generate an orthotopic xenotransplant, as we already did with success for other very rare soft tissue sarcoma, such as, for example, desmoplastic small round cell tumor. How can we exploit PDX for translational research in EHE? Our generated PDX model is relatively recent, so we did not perform many experiments thus far. However, we starting focusing on 2 different aspects. One is the use of this model to analyze drug activity, and the second one to try to identify and preclinically validate novel biomarkers for the disease. As far as drug activity is concerned, we started to compare the activity of sirolimus, that is a drug quite active in this disease, with the golden standard drug for soft tissue sarcoma, that is doxorubicin. We started by analyzing the cytotoxicity of the two drugs in our cell line, and we found that sirolimus started to inhibit cell growth at concentration lower than those required for doxorubicin, as demonstrated by the 50% inhibitory concentration that is 1 log lower for sirolimus compared to doxorubicin. However, sirolimus quite soon reached a plateau of the activity, suggesting that its activity is more cytostatic than cytotoxic. And this was also confirmed when we look at the percentage of apoptotic cells following treatment, that was very low, as determined by flow cytometry as percentage of tunnel positive cells. Then we move in vivo in our PDX model. Also in this case, we compare the activity of doxorubicin to that of sirolimus. For doxorubicin, we use the standard schedule and dose that is used in almost all the study on tumor xenotransplant. For sirolimus, we use two different doses that were also derived from the literature in experimental model of other tumor types. As you can see, the model was completely resistant to doxorubicin, the red line. On the other hand, sirolimus induced a very good anti-tumor activity in these models, but the activity was not dose dependent. Indeed, we observe a max tumor volume inhibition around 80% by using both the doses. As you can see, in the case of sirolimus, we do not have a reduction of the tumor volume, but we have a stabilization of the tumor. This also confirms that the effect of the drugs is mainly cytostatic. Then, at the end of experiment, we remove the tumor from untreated and treated animal and look at the proliferation index in this tumor by determining the Ki67 index. As you can see, the Ki67 index in doxorubicin treated tumor is only slightly lower than that observed in control untreated tumors. But the Ki67 index in sirolimus treated tumor was much lower and significantly different. In order to see how was possible to demonstrate the known mechanism of action of sirolimus in our model, we perform Western blotting analysis in one PDX untreated and in 4 PDX treated with sirolimus. And as you can see, in all the cases, there was a complete abrogation of the signaling sustained by mTOR. The results we obtained thus far with drugs indicate that this model is suitable to analyze the activity of drugs. Indeed, we are already planning to use this model to analyze more novel drugs, such as, for example, drugs that interfere with the YAP-TATS pathway. As I told you before, we thought to use our patient-derived models also to identify novel biomarkers for the disease. In a studies that is carried out in my lab, we look at possible circulating biomarker for the disease, and particularly we focus on circulating cytokines. By comparing the circulating cytokines in the blood of EHE patients and of healthy donors, we found a certain number of cytokines that are differentially expressed. As an example, I will show you our data on the growth and differentiation factor 15. this factor was upregulated in the blood of EHE patients. And when our oncologists subdivided the patient into different categories according to the extent of clinical aggressiveness of the disease, we observed that the higher level of GDF15 expression were observed in patients carrying the most aggressive disease. So we were interested in see how the tumor contribute to GDF15 expression in the blood. For doing that, we use our cell lines, plate the cells, starve the cells in absence of serum, and then look at the conditioned media to see the protein that are released from the tumor. To assess the cytokine profile, we use a cytokine array, and we were able to see that among the released cytokine, there was the GDF15. The next question was to see how sirolimus could interfere with the release of GDF15. So we measure the expression of the cytokine in the medium of control cells and of cells treated with sirolimus, and normalized the expression by the number of cells. And as you can see in the picture in the bottom part of the slide, sirolimus was able to reduce the release of this cytokine. So we thought to challenge our in vitro data in the in vivo models. For doing that, we look at the circulating level of GDF15 in 3 healthy mice without the tumor, as well as in 2 mice nominated PLS carrying PDX of pleomorphic liposarcoma. And in all these mice, it was not possible to measure any amount of GDF15 in blood. In the other end, all the mice carrying the EHE PDX release in the blood a certain level of GDF15. Also in this case, we wanted to see whether sirolimus was able to downregulate the release of GDF15. And we express the quantity of circulating GDF15 in control mice, and that observed in the blood of mice treated with 2 doses of sirolimus. As you can see, there is an enormous difference that cannot be explained by the different volume of the tumors. In our institute, there is an ongoing study in which we collect longitudinal samples of plasma from patients who are undergoing treatment with sirolimus in order to see if this data can be confirmed also in humans in order to exploit a possible clinical relevance of GDF15 as a novel biomarker for monitoring drug activity. In conclusion, I must say that the preliminary results that we obtained thus far on our EHE PDX models indicate that this model mainly recapitulates the characteristic of the clinical originating tumor. It seems to be suitable for the assessment of the activity of drugs and could provide important information for the design of novel therapeutic trials with a strong preclinical background. And the model seems to be also useful for the identification and preclinical validation of novel biomarkers. I would like to thank all the people belonging to the sarcoma team of INT that collectively work in all the translational research studies in my institute, and of course the EHE Rare Cancer Charity UK, as well as the EHE Foundation USA, and you for the attention.
All right, it's time for a roundtable Q&A. I'd like to invite back Dr. Al-Jazrawe, Seavey, and Zaffaroni, as well as bring Dr. Rubin in for a group discussion here. We do have a few questions that have come in from the attendees. The first one is for Dr. Al-Jazrawe. In your experience with other cancers, can you use transgenic mouse models to test different isolation culture conditions and apply those to human samples of the same cancer type? And I guess the issue here is more availability of tissue and how well that would correlate.
Yeah, I think that's a great question. So far, we haven't tried any engineered cells or animal cells, although the concept is the same, that we want to use this technology for barcoded sequencing to systematically address how molecular features relate to what culture conditions or ex vivo conditions can help growth or hurt growth. So you can potentially ask that, hey, we can try different engineered cells, be it from mouse or human, and then we can try and see, what supplements are helping a specific allele to dominate the culture, so to speak. But we haven't really tested any. So far, we focused on the human patient samples we received.
Thank you. And we've got a question for Dr. Seavey. Have you knocked down TAS-CAMTA1 in your MEHE cell line to see if growth in culture or in vivo are still dependent on the fusion gene?
Yeah, it's a very, very difficult. Can you hear me?
Yes.
Okay. That's a very, very difficult question. We've tried it multiple times. We're not getting an adequate knockdown in multiple of our experiments. Our suspicion is it's probably because it's so essential for its proliferation and for its maintenance. So we're right now pushing ourselves toward really, really early experiments where we're trying to knock it down and then immediately start to assay whether there's alterations in proliferation and does it increase apoptosis? So we're doing a lot of that work, but to generate stable cell lines of TAS-CAMTA1 knockdowns has not been successful, and I suspect it's probably because it's still oncologically addicted to TAS-CAMTA1, and that's true for multiple of our cell lines.
Thank you much. And I've got a question for Dr. Zaffaroni. The question reads as follows. It is well known that mTOR inhibition results in a feedback activation of AKT. Do you know why sirolimus decreased AKT phosphorylation in your cell line?
Yeah, I have seen a decreased AKT phosphorylation in our PDX models, but different results have been observed in the cell line. So we are going in deep into the questions to see what happens.
Thank you much. And then we have one question that actually came in from the last session, but it's not tied to any particular model. So I'd throw this out to anyone in the group who's able to answer it. Since doxy induces gene expression, has anyone looked at the frequency of prior doxycycline or tetracycline exposure in patients with EHE? I'm seeing shaking heads now.
No, I'm guessing.
All right, that takes the questions from the audience. I have a couple, but I'll see if Dr. Rubin has any questions or thoughts listening to these talks.
I don't have any questions, but it's just nice to see so much modeling that, we're starting to see some success here. And so kudos to everybody who just presented. I know how difficult these models are to produce. And, Caleb's done a wonderful job in my lab. I mean, Nadia's xenograft / cell line shows a lot of promise. Everybody else who's doing unusual things, trying to express in embryonic stem cells. I think that's an interesting idea. And of course, Freek with the Zebrafish model. I think all these things could be important. They're all going to answer different aspects and be useful in different ways. And it's wonderful to see all this progress.
Yeah, and the thing that seemed exciting to me is the diversity of the models here when we get into testing drugs really is going to be beneficial, right? Because some of these models are going to be more amenable to different types of or classes of drugs and different type of screening approaches. So this really opens up the possibilities. A question for Dr. Zaffaroni on the GDF15. Is that shown up in other cancers as an interesting marker, or is this EHE unique?
The role of these cytokines in cancer is very controversial in the sense that its expression is very low or does not exist in normal tissues, but it seems to be upregulated in certain cancer and also in cancer in tumor microenvironment. But there are different way in which this cytokine can participate in tumorigenesis. It seems to have opposite function, pro-tumorigenic or anti-tumorigenic, depending on the tumor type and also on the stage of the disease. But thus far, there are no, let's say, relevant information for soft tissue sarcoma in general. So it's interesting to see. This GDF15 is also being proposed as a potential therapeutic target, for example, in glioblastoma multiforme. And we are looking in our model whether, for example, downregulation of its expression by siRNAs or short terpene RNA can influence the growth in vitro and the tumorigenesis in mouse.
Excellent. And let me turn it open to the panel here in general. Does anyone have any questions for each other? All right, if not, we can close up this session. We're going to take a 10-minute break. We're a little off the normal schedule, but we will have this 10-minute break, which I think everyone will need, and then join into the afternoon session after that break. Thank you again for everyone's time today.